Hello guys, so welcome back. So today we shall be discussing another important topic of your enterobacteria, that is Campylobacter jejuni, right? So that topic is called as that bacteria is called as Campylobacter jejuni. Now, while discussing Campylobacter jejuni, first of all let us discuss the general properties of this bacteria and then we shall switch on to the next things, okay? So first of all let us discuss about the general properties of the bacteria or the general characteristics. So, when it comes to the general properties of the bacteria, can you tell me the first thing we have to start with? We have to start with whether it is a gram positive or a gram negative. How are we going to know whether a bacteria is gram positive or negative? By looking at the peptidoglycan layer. And this bacteria is having a very thin peptidoglycan layer. So, I call this bacteria as gram negative bacteria. I call this as gram negative bacteria. Second important thing is regarding the properties, right? So this bacteria can release an enzyme called as cytochrome oxidase. So it is called as oxidase positive bacteria. It is called as oxidase positive bacteria. Third important thing is that when you look at this bacteria, right, I'll show you the picture of bacteria in a minute. But when you look at this bacteria, you will find a flagella, right? So can I call this as a flagellated organism? This is a flagellated organism, right? So if an organism is flagellated, so what will be its property, guys? What will be its function? Function is that it is responsible for motility. It is responsible for motility. Now, if you ask me, one important thing that I have to discuss about this bacteria is the most common cause of gastroenteritis is Campylobacter jejuni. Okay, so most common cause of gastroenteritis. Most common cause of gastroenteritis is this your Campylobacter jejuni. This is the most common cause of gastroenteritis. Okay, and by the way, when you look at this bacteria is not straight, rather this bacteria is little bit curved like this. Okay, so it is a curved, and if you exactly look, it is S-shaped bacteria. So it is an S-shaped curved bacteria. Okay, so Campylobacter jejuni is an S-shaped curved bacteria. It is an S-shaped curved bacteria. Now, another important thing which you need to understand is that, for example, if we discuss about how this bacteria is transmitted to you, right? So let us discuss about the transmission of this bacteria. Let us discuss about the transmission of this bacteria. So all of you have to know the transmission way is obviously when you are eating, when you are drinking milk, what type of milk? Unpasteurized milk. Not only unpasteurized milk, any kind of unpasteurized dairy products. Okay, so unpasteurized milk, improperly cooked or uncooked poultry, right? So uncooked poultry or unpasteurized milk, so any one of these can be a potent transmission factor. Okay, so it can be unpasteurized milk or it can be uncooked poultry. So this can be a potent transmissional factor, right? So obviously you ingest them. So the main route is fecal oral transmission. Fecal oral transmission. So fecal oral route of transmission. The risk factors are unpasteurized milk and uncooked poultry. So let us discuss about the pathogenesis of how this bacteria mainly affects your intestine, leading to gastroenteritis. Okay, so let us discuss the pathogenesis. In the pathogenesis part, you need to know one thing. What is that thing? Where is the colon here? Look here. This colon which I am drawing right now in front of you, right? So this colon is called as your ascending colon, and this one is called as your transverse colon, okay, and this part is called as your descending colon. So what are the different types of colons you have, guys? You have got ascending colon, transverse colon and descending colon, and at the end you have got this type of part here. This is called as sigmoid colon, which is S shaped, right? And after that you have got your rectum and anus. This thing you learn in your anatomy. But what you need to know here is that, guys, what you need to know here is Campylobacter jejuni comes and resides within your large intestine. So when it will reside in your large intestine, how is it going to affect you? How is it going to affect you is that, look here, there are few cells here, okay? There are few cells. These cells are called as enterocytes. All these cells which I am drawing here, these blue color cells, right? So let me make a partition, right? So these are the blue color cells which I am drawing here. These are enterocytes. So what I am going to represent on my right hand side is that I will draw one very big enterocyte, and in front of that I will draw the bacteria, and let us see what is happening. But before this you need to know what are the different layers of your intestine. Tell me, what are the different layers of your intestine? The first layer if you see here, the first layer in the sense, for example, see, this is a colon, okay? Now I have cut the colon. See, this entire pipe which I am representing here, from this region all the way down, this is your colon. This entire thing is your colon. Now if I am looking into this colon, I will find a lumen. If I am showing it to you, I will show it like this. So all this part is the lumen. So whatever thing that is lining the lumen like this, right? Look here, all this part that is lining the lumen, here also, the inner cavity that is lining the lumen, this is coated by a mucus layer. All of you know that, because mucus is the first barrier. So it is coated by a mucus layer. So can I call the first layer as mucosa, right? So the first layer will be mucosa. Okay, so after mucosa, the next layer, surrounding mucosa, you have got the second layer, that is called as submucosa. The next layer is called submucosa. Obviously in mucosa you also have 3 more layers. We are not going into that much detail now, but what you need to know is, after mucosa you have got submucosa. Surrounding the submucosa you have got muscularis externa. Surrounding the muzzle layer, surrounding the submucosa, you have got muscularis externa. And surrounding that muscularis externa, finally, you have got your what? You have got your serosa or adventitia. Serosa or adventitia. So these are the layers from inside to outside. Right. So let me explain you in another way. I hope all of you understood, but for those who did not understand, what I am trying to tell you is that, see here, this thing which you can see here, this is the innermost layer. You see this? You see a cavity inside, this is called as a lumen, okay? Now surrounding this, I have got my second layer that is surrounding this like this. I have got my second layer that is surrounding this, that is called submucosa. Now surrounding that submucosa, I have got muscularis externa, that is a muzzle layer. We need the muzzle layer for the peristalsis of your intestine, right? For the peristalsis of your colon, obviously you need it. Surrounding that, the outermost layer is called as your serosa. So what is the main reason that I am explaining this to you is that, in the submucosa layer, you have got blood vessels like this. In the mucosa layer, you don't have any blood vessels. In the submucosa layer, you have blood vessels. For example, if the infection is only in the mucosa layer, nothing is going to happen to you. But if the infection goes deep inside the submucosa layer, it will rupture the blood vessels, leading to bleeding. Okay. So let us discuss that. Okay. Let us discuss the mechanism now. Now, first of all, our main agenda is that we have to attach this bacteria to your enteric cells. Now, how do you attach this bacteria to your enteric cells, guys? Look here now. Let us say this thing is your bacteria, and I already told you bacteria has got a flagella, right? So let me write it down. Let me draw it down that this is the flagella, maybe. Okay. Now, surrounding the bacteria, I am not going to draw the structure of bacteria in detail now. Surrounding the bacteria, you need to know that there are some small structures like this. Okay. There are some small structures like this. Now, along with these structures, there are some more structures in the form of chains. You see, in between these small structures, hairpin-like structures, there are some more structures in the form of chains. Now, what are these two structures here? What are these two structures is that these small structures which you can see here, this is called as CAD-F molecule, CAD-F protein or CAD-F molecule, and the long chain like structures which you can see here, they are called as PEB1 molecule. Okay, one is called as PEB1 and the next one is called as the CAD-F molecule. CAD-F molecule. So why do we need them? We need them for the proper attachment to the host cell. Okay, so if this is my enterocyte over here, if this is my enterocyte over here, see the bacteria is very properly attached to my enterocyte. And how did it attach to my enterocyte? With the help of this CAD-F as well as PEB1 molecule. Okay, so can I tell PEB1 and CAD-F molecule are responsible for adhesion? So can I call both of them are adhesins? Yes, I can call these both molecules as adhesins. So can I call these both molecules as virulence factors? Yes. Any factor that is, for example, if someone is committing a crime, the person who is committing a crime is the culprit, and the person who has given a plant for him to commit a crime is also a culprit. Every individual who is involved in that crime is a culprit. The same thing, everything that is bad happening to your body, because of either it might be adhesins, adhesins might not release any toxin, but they are responsible to stick to the host cell, right? So in some or the other way, they are also involved in the inflammation. So I can call them as virulence factors. Anything that is responsible for the damage of your body is called as a virulence factor. Okay, fine. So these are called as adhesins. Now let us discuss how the things happen here. Let me draw the enterocytes here. Okay, so let me draw it down over here. So this is your enterocyte. And where are these enterocytes located? They are located in the mucosa layer, right? So mucosa is the innermost layer. Now this is the mucosa layer, the innermost layer where enterocytes are located. Next to that I have submucosa. It means, look here, all this layer here is called as okay, instead of drawing the lines, let me draw the dotted lines. Okay, let me draw the dotted lines so that it would be clear for you. So all these layers which you can see here, this entire lines which I am drawing right now, just be patient for a second. So all these lines which are drawing right now, all this is called as the submucosa layer. Okay, so what is this layer called as, guys? This layer is called as a submucosa layer. What is the importance of submucosa layer? Obviously submucosa layer has got blood vessels. Okay, submucosa layer has got blood vessels like this. So there is extensive branching in this way. Submucosa layer has got blood vessels. So what is this layer here? This is called as mucosa layer, and the next layer is called as submucosa layer, right? So we have got our mucosa, we have even got our submucosa layer. Cool. Now this was our bacteria, right? So this was our bacteria. Now this bacteria, I told you, it was having what on the back? It was having its flagella on the back, right? So finally this bacteria very firmly attached to the enterocytes. Now it attached to the enterocytes with the help of adhesion molecules. Fine, that's cool. Now how is it going to enter inside? Attachment is okay, but how is it going to enter? It is going to turn its side back. It is going to turn back, and on the back side it has got its flagella. It will drill the enterocyte with the help of its flagella. You see, let us say this is the enterocyte. Now this is the flagella. Now the flagella is attached here. Now look, the flagella will drill it like this. It will drill and finally it enters inside. The bacteria enters inside. So it drills the flagella in this way and finally the bacteria is in. It drills the flagella and finally the bacteria is in. Now this bacteria here, guys, it multiplies. You know, this bacteria will multiply in the enterocyte like this. This bacteria will multiply in the enterocyte in this way. Okay. So by the way, it is multiplying in the enterocyte like this, and after multiplication it will start releasing a toxin. It will start to release a toxin, and this toxin is called as cytolethal distending toxin. This toxin which it has released just now, guys, this toxin is called as cytolethal distending toxin. This is called cytolethal distending toxin. What is the name of this toxin? Cytolethal distending toxin. Okay. Now what this cytolethal distending toxin will do? It will cause severe inflammation of your enterocytes. Okay. It will cause severe inflammation of your enterocytes. The moment this toxin is released, guys, obviously your immune system is not silent. Then immediately all your immune cells, every part of your immune cell. Okay, let me tell you in detail what is happening. Okay, let me tell you in detail what is happening. The simple thing is that, guys, in this cell, in this particular cell, we have got our, guys, what is that name? We have got our macrophages. So, you see, these are the macrophages. Okay. So what these macrophages will do is that these macrophages will engulf those toxins. I mean this cytolethal distending toxin. Okay. So macrophages, it will engulf those toxins. See, macrophages have engulfed some toxins. After engulfing the toxins, these macrophages, they release two very important things. What are these two important things? What do they release? They release interleukin 1, interleukin 8. What did they just release, guys? They release interleukin 1 and interleukin 8. And what is interleukin 1 responsible? I have taught you this in the previous lectures very clearly. But again, I am telling you that remember that interleukin 1 is responsible to cause fever in your body. And interleukin 8 is responsible to cause inflammation in your body. Inflammation. So one is responsible for fever and the other is responsible for inflammation. So excess amount of toxins, for example, let us say there is heavy concentration of this cytolethal distending toxins within the cell. Then obviously lots and lots of macrophages will come there. And lots of these macrophages, they start releasing excess amount. The more the toxins, the more the release of interleukin 1 and interleukin 8. These are the cytokines, right? So excess amount of release of interleukin 1 and 8. So patient will have high fever and heavy rate of inflammation. Patient will have heavy rate of inflammation. Okay, right. So patient will have excess inflammation. And where is this all happening? This is happening in the colon. So in the colon, can I tell all the enterocytes are damaged now? Yes. Can I tell that? Yes. These toxins, they damage the entire enterocyte. So now look at the picture of the enterocyte, what it is going to become next. This enterocyte will become like this, floppy. The enterocyte is modified into a floppy form, floppy enterocyte, right? And slowly, you know what will happen? Slowly, the enterocyte will become black. This is called as necrosis of the enterocyte. Okay, necrosis of the enterocyte. And in the later on stages, you know what is going to happen? Later on stages, look at the picture of the colon now. This was your colon, right? This is your transverse colon, like this. This is your descending colon. So what has happened? What has happened is that all your enterocytes became floppy. You see, all the enterocytes are completely compressed and dead like this. All these enterocytes are almost compressed and dead like this. So in this case, what will happen is that, guys, there is severe inflammation that is right now going on in your colon. So colon will be so much inflamed that colon will distend out. Moreover, the toxin which it released is cytolethal distending toxin. So it will cause complete distension of colon. So colon slowly, as the inflammation is increasing, colon slowly distends, and finally, if you look at the picture of colon, this is how the colon looks. This is your ascending colon, transverse colon, and this is your descending colon. You see how the colon looked? Those markings which you see between in the colon, these are called as haustra, right? So these are called as haustra. I mean, that projections inside are called as haustra. Now tell me whether you find haustra here or no. There is no haustra, right? There is no haustra. Why do you think that the colon has distended? I told you already that this bacteria released a toxin that is called as cytolethal distending toxin that caused severe inflammation and distension of all the cells, and this led to the distension of colon, right? Second reason is that after submucosa, what is the next layer, guys? Muscularis mucosa, right? After submucosa, we have got muscularis mucosa, where I have muscle layers. You know, I have two types of muscle layers. Look here. These muscles which I am drawing now, these are called as circular muscles. And these muscles which I am drawing now, these are called as longitudinal muscles. Okay. So circular muscles and longitudinal muscles, together I call them as muscularis externa. Together I call them as muscularis externa, right? So muscularis externa is comprising of what? Is comprising of circular muscles inside and longitudinal muscles outside. So I was telling you that this inflammation is too much, that this inflammation can even enter into the submucosa, okay? And from the submucosa, the inflammation can even enter into the muscularis externa, the muscle layer, and can damage the muscles. You know what will happen when the muscles get damaged? They become paralyzed. When the muscles are damaged, they no more work. Okay. So, when the muscles of the colon are damaged, look in the picture now. Okay. So, let me draw, see this red color thing which I am drawing is the muscle layer. Okay. This is the muscle layer on both the sides. And this infection even entered into the muscle layer and caused damage of the muscle. So, let me remove the muscle layer here. Removing the muscle layer, which means there is damage of the muscle. So let me write it down here, you know, this is damaged muscle or inflamed muscle. Now when the muscle is damaged, do you think will it work? No, it won't work. So can it perform this function, that is peristalsis? No. So when it cannot perform peristalsis, do you think that the food which is present in the colon, can it be propelled forward? Can there be peristalsis of this food in the forward direction? No. So when the muscle layer is damaged, there is no contraction, there is no peristalsis. So the intestine remains in this way still, right? So the intestine remains in a standstill position. So there is stasis of the intestine, there is no peristalsis. So even the food cannot move forward. So when the food cannot move forward, the food starts accumulating and leading to a distended colon. And this colon which I have written here in front of you, this is called as toxic megacolon. So this colon, guys, is called as toxic megacolon, right? Next thing what can happen, guys? Next thing what can happen is, before happening toxic megacolon happens when the muscle layer is damaged. But before that you have got submucosa, right? But by chance, if these toxins, they enter into the submucosa layer like this, and from the submucosa layer you have got blood vessels. So it will even enter into the blood vessels like this. So when the toxins enter into the blood vessels, what will happen? There will be bacteremia. Yes or no? There will be bacteremia. So if there is excessive bacteria within the blood vessel, then what will happen? That will lead to what? Sepsis. That will lead to sepsis in the patient. Okay, right. So what are the three most important things? What are the things that are happening now, guys? One is bacteremia. Second one is patient will have fever, right? Third thing is patient was having inflammation. Obviously the entire bacterial discussion is about inflammation. The fourth important point is it can even cause toxic megacolon. Toxic megacolon, right? So these are the most important things which you need to know, right? So what are the conditions, what are the diseases that are associated with this Campylobacter jejuni? Look here now very carefully. Campylobacter jejuni, okay? So this Campylobacter jejuni is associated with three most important diseases, guys. What are these three most important diseases are? The first important disease is gastroenteritis. I already told you most common cause of gastroenteritis is Campylobacter jejuni. So it is associated with gastroenteritis. It is associated with what? It is associated with gastroenteritis. If a person is, obviously now you can't ask me that how gastroenteritis. I have explained you the entire mechanism. When all that mechanism is going on there, don't you think the intestine is damaged now? The intestine can no more absorb any nutrients or water or anything, right? Look here, guys. Look here now. All the intestine is damaged. You see, all these cells became dark. They became dark. They are damaged now completely. So when your entire intestine is damaged and when your entire intestine is under heavy inflammation, do you think so ions like sodium, potassium, chlorine, right, water, can they be absorbed into the enterocytes? No enterocytes itself. How do you think that from the lumen all these electrolytes will be absorbed into the cells? Impossible. So this absorption which was happening will not happen now. So what will happen now? What will happen is, as the cells are not in a state to receive sodium, receive potassium, receive water, all the sodium, potassium and water along with the chlorine, they get excreted out. So that will cause watery diarrhea in a patient. But if the patient is not getting any treatment, okay, let me write it down first thing. Patient will have what type of diarrhea? Patient will have, obviously patient is going to have watery diarrhea. Not only watery diarrhea. Later on what will happen is that this watery diarrhea will progress to bloody diarrhea. How? How, tell me now? Look in the picture now. If the inflammation is within the cell, only if the cell is dead, only if the cell is damaged, then only if the cell is damaged, then the sodium ions, the potassium ions, the chlorine, the water, all these three things, the sodium, the potassium, the chlorine, the water, they cannot enter now. They cannot enter. Instead, they will be excreted out. They will be excreted out, right? So they will be excreted out. So when all this watery diarrhea thing is happening, it is happening when only the cell is damaged. But if the infection will enter little more inside the submucosa, then it will damage the blood vessels leading to hemorrhage, right? So along with this watery diarrhea, blood also, I mean hemorrhage, you know what is hemorrhage, right? There is breakdown of the vessels. So blood, along with the blood, the watery diarrhea and blood together they form and lead to bloody diarrhea. So Not only watery diarrhea will progress to bloody diarrhea. Watery diarrhea is going to progress to bloody diarrhea. So when all that thing is happening in the patient, don't you think patient will have abdominal pain? Obviously, patient will have abdominal cramping. Abdominal cramping pain. Okay. Next, apart from gastroenteritis, can you tell me another thing that can happen to our patient? Yes. Apart from gastroenteritis, patient can also have a syndrome, right? That it is believed that this toxin will demyelinate all the neurons and it will lead to a syndrome that is called as Guillain-Barré syndrome. Guillain-Barré syndrome. It will lead to a syndrome called Guillain-Barré syndrome. And the third important thing, you will learn about Guillain-Barré syndrome very much in detail in physiology as well as in your internal medicine, right? So even I would like to teach you about Guillain-Barré syndrome now, but that is not necessary for your microbiology. Okay, no one is going to ask you in your MBBS exams what is Guillain-Barré syndrome in your microbiology especially. So the third important thing is that patient can also have a reactive arthritis. Patient can have reactive arthritis. So these are the three most important things which one has to know. One is gastroenteritis, Guillain-Barré syndrome and reactive arthritis. Everyone have to know these things. Okay. Right guys, so what is reactive arthritis? In reactive arthritis, keep one thing in mind, patient can't see, patient can't pee, patient can't bend. Patient can't see, patient can't pee and patient can't bend. Patient can't see, problem with the eyes, patient might have uveitis. Patient can't pee because problem with the urine, patient will have urethritis. And patient can't bend the knee because he will have arthritis. So, reactive arthritis is a triad. Reactive arthritis is a triad of all these three things which you have to know, right? So, thank you so much.